Assalamu alaikum students, I am Dr. Sadia from Pathology Department. I will start a new topic genetics today, and in genetics I will talk about mutations and mode of inheritance. Today I will discuss different types of mutation, modes of inheritance, transmission pattern of single gene and sex-linked disorders. All of you know DNA is a double-stranded structure which is held together by hydrogen bonds between different bases. So replication is the process of formation of DNA from DNA, while transcription is the process of formation of RNA from DNA, and in translation proteins are formed from RNA. You can see difference between DNA and RNA. RNA is a single-stranded structure while DNA is double-stranded structure. There are different types of RNA, ribosomal RNA, transfer RNA, and messenger RNA. All of them play important role in protein formation. Here are some definitions. Genetics is the study of single or few genes and their phenotypic effects, while genomics is the study of all genes in the genome and their interactions. Proteomics concerns itself with the measurement of all proteins expressed in the cell or tissue. To analyze pattern of expression involving thousands of genes and proteins has required the parallel development of computer-based techniques called bioinformatics. Epigenetics is defined as heritable gene in gene expression that are not caused by alteration in DNA sequence. Epigenetic changes are involved in tissue-specific expression of genes and genomic imprinting. DNA and RNA are genetic blueprints. Both are nucleic acid. DNA carries genetic code while RNA transcribes genetic code and play important role in protein formation. Here you can see process of formation of protein. mRNA is transcribed from DNA and then it's translated to form a polypeptide. What are genes? Genes are sections of DNA that determine certain traits or characteristics. Different versions of the same genes are called alleles. Gene can have two or more possible alleles. You can see here this section of the chromosome specifies the trait for eye color, while this section of chromosome specifies the color, hair color. So this is gene. While alleles produce diversity in eye color. So alleles are alternative forms of gene. Here you can see different hair color will be determined by alleles. Human gets 2 copies of every gene, 1 copy from each parent. The 2 copies don't have to be identical. For example, if a gene contains information for eye color, one allele might code for blue eyes and another might code for brown eyes. Principle of dominance. One gene may mask another gene when 2 are found together on a heterozygote. Dominant gene's characteristics are observable, while concealed gene is called recessive gene. Gene for a recessive occur in heterozygote who carry 1 copy of the gene but do not express the disease. Dominant allele is one that always determines the phenotype when present. On the other hand, recessive is one that is not expressed when its paired allele is dominant. For example, with the eye color. The brown eye allele is dominant to blue eye. This means that a child with blue allele from their mom and brown from their dad will end up in brown eyes because brown color is dominant. But a child with 2 blue alleles will display the blue phenotype because both are recessive genes. Codon is a 3-base sequence. You can see here different codons composed of 3 base pair sequence. Regulatory function of microRNA are special RNA that are coded by specific gene on a chromosome. First, prime microRNA is formed, that is converted to pre-microRNA, that is transported out of the nucleus to form pre-microRNA. Dicer enzymes cleaves pre-microRNA into microRNA. Then unwinding of microRNA occurs and a microRNA is incorporated into ribosome-induced silencing complex. This complex is very important. It can attach to the target mRNA, either cleaves it or translational replacement. This is how gene silences occur at post-transcriptional level. Genetic disorders. There are different types of disorders related to single gene with large effect, chromosomal disorders, or complex multigenic disorders. Mutation is the change in DNA sequence. This change may affect a single gene or an entire chromosome. There are 5 types of chromosomal mutations that occur: deletion, inversion, translocation, nondisjunction, and duplication. Deletion is due to breakage. A piece of chromosome is lost. You can see pink portion is deleted and the length of chromosome is shortened. While inversion, chromosome segment breaks off, flips around and reattaches. You can see here the chromosome shown in pink and green color. It breaks off, flips over and then reinserted. Duplication occurs when a gene sequence is repeated. Here it's shown in yellow color. Yellow part is duplicated and the length of chromosome is increased. Translocation involves 2 chromosomes that aren't homologous. Part of one chromosome is transferred to another. Here they are shown in black and white color. The black part is transferred to the white chromosome. This process is called translocation. Nondisjunction, failure of chromosome to separate during meiosis, causes gametes to have too many or too few chromosomes. For example, in Down syndrome, patients have 3 21st chromosomes, while in Turner they have single X chromosome, and in Klinefelter again they have double X chromosome. Here you can see the process of non-disjunction. Here tetrad can't separate, and after meiosis too, either the cells are having one extra copy of chromosome or one less chromosome. Here you can see examples of chromosomal mutation. Here purple and green part is repeated, duplicated. This is the example of duplication. The next is the example of deletion. Purple part is deleted. This is the example of inversion. They flips over. And translocation is the transfer of section of chromosome between 2 different chromosomes. Mutation may occur in somatic cells and passed to offspring, and that may occur in gametes. Eggs and sperm can be passed to offspring. There are different types of mutation: gene mutations and chromosomal mutation. In gene mutations, we have point mutations and frame shift mutations. Point mutations are change of a single nucleotide. It includes deletion, insertion, or substitution of one nucleotide in a gene. There are different types of point mutation: silent, that code for same amino acid; missense, that code for different amino acid; nonsense mutation, which code for a stop codon. Other includes deletion, substitution, and insertion. They cause frame shift mutations. Here you can see example of different mutation. At DNA level, lysine is coded by TDC, while if one base is changed and still lysine is formed, this type of mutation is called silent mutation. While if the mutation causes conversion of normal codon to stop codon and stop the process of protein formation, it's called nonsense mutation. And if codon code for another amino acid instead of lysine, like arginine or threonine, it's called missense mutation. Missense mutation is of 2 types, conservative and non-conservative. If the new amino acid is similar in physical or functional properties to the required amino acid, is called conservative. While if amino acid belongs to other chemical group, it's called non-conservative. This is the example of sickle cell disease in which beta globin chain synthesis is abnormal due to one nucleotide change that results in valine formation instead of glutamic acid. This is the example of missense mutation. What you ordered, you are not received. In sickle cell anemia, valine is produced instead of glutamic acid. That results in sickling of the RBCs under the effects of hypoxia. Sickle cell disease is the result of one nucleotide substitution occurs in the hemoglobin gene. Here you can see sickle cell formation in hypoxia. Cystic fibrosis, broken salt channel in cells, strikes in 1 in 2500 white bloods. Gene codes for a protein channel that allows salt to flow across cell membrane. Broken protein doesn't work as a channel, doesn't allow salt out of water, so water doesn't flow out. Thicker and stickier mucus coating around the cells cause mucus buildup and cause bacterial infection. Without treatment, child die before 5. With treatment, they can live past their 20s. Here you can see this channel cause salt transfer to the lumen. Water goes under the effect of osmosis. Normal mucus is produced. Salt is not exported out. Osmosis doesn't cause more bacteria to grow. Frameshift mutation. Inserting or deleting one or more nucleotides changes the reading frame like changing a sentence. Proteins built incorrectly. Here you can see if the sentence is the fat cat ate the rat. If we insert one H here, one nucleotide here, all the sequences change because codon is a 3-base sequence, so all the reading frame is changed. While same is the case with deletion, if you delete H here, all the reading frame is changed. Due to triplet nature of codon, gene expression by codons, the insertion or deletion can disrupt the reading frame, the grouping of the codons resulting in a completely different translation from the original. You can see if C is deleted here, all the sequence of amino acid is changed. Again here, if glutamic acid is deleted here, guanine is deleted here, so the result is formation of different amino acids because all the reading frame is changed. Original is the fat cat ate the wee rat. So if we add A here, this is the example of insertion of one nucleotide. If we add A here, all the reading frame is changed and it becomes fat cat ate you. This sentence is meaningless. Causes of mutation may be inherited from parents to child or acquired mistakes when DNA is copied. May be due to oxidative damage. Cytosine and thymine are more vulnerable, while pyrimidine dimer are formed by ultraviolet light. Chemicals can also cause mutations like hydroxylamine, base analogues, and alkylating agents. Most of the mutations are neutral, like eye color and birthmarks gets unnoticed. Some are harmful, like sickle cell anemia and Down syndrome. Some are beneficial also. For example, sickle cell anemia patients are resistant to malaria. And some of the defects in receptor cause immunity to HIV infection. You can see example of aniridia with PAX6 gene. Snake with 2 heads, duckling with 1 more extra part. These are all examples of mutation. Animals with extra body parts. Web2 that occurs in Anderson syndrome is also an example of mutation. Black jaguar, a gene mutation overproduces melanin in the fur. Belgian blue bull due to gene mutation for a muscle growth that cause it to happen without any steroids. So we discussed point mutation, simplest change, one nucleotide is exchanged for other. In missense mutation, changes the triplet codon making a different amino acid. Frame shift, 1 or 2 nucleotides are inserted or deleted, which changes all codons beyond that point. Nonsense early stop codon that cause stop of protein formation. Regulatory change in a sequence important to make proteins. Patterns of inheritance. We will discuss here Mendelian inheritance that may be autosomal or sex-linked, chromosome-linked. Both are dominant and recessive. Transmission pattern of single gene disorder. Autosomal dominant, recessive, and X-linked. In autosomal dominant disease, only one mutated copy of the gene will be necessary for a person to be affected by autosomal disorder. Each affected person usually has one affected parent. The chance a child will inherit the mutated gene is 50%. Both males and females are affected equally and both can transmit disorder. They often involve dysfunctional receptors and structure proteins. Late onset disorders. Affected individuals may remain asymptomatic till adult life, as in Huntington's disease. Variable expressivity. Difference in the severity of expression. For example, neurofibromatosis patient may present as Brownie spots, or some may present with multiple tumors and skeletal deformities. Reduced penetrance. Some persons inherit the mutant gene but are phenotypically normal. New mutations also occur, involving either egg or sperm from which they were derived. Their siblings are neither affected nor at increased risk for development of the disease. Here you can see affected father can transmit disease to 2 out of 4 of the children. So the next child will have 50% of chance to be affected. Examples include Huntington disease, neurofibromatosis type 1 and 2, Marfan syndrome, and others. Autosomal recessive disease. 2 copies of the gene must be mutated for a person to be affected by autosomal recessive disorders. Usually have unaffected parents who each carry a single copy of mutated gene and are referred to as carrier. 2 unaffected people who each carry 1 copy of mutated gene have a 25% chance with each pregnancy of child having affected by the disorder. You can see both parents are carrier of mutated gene, so they have 1 out of 4 child to be affected, and 2 of their children will be carrier. The expression tends to be more uniform than autosomal disorder. Complete penetrance is common. Onset is usually early. Although new mutations occur, they are rarely detected clinically. Examples include phenylketonuria, homocystinuria, galactosemia, glycogen storage diseases, and others. Sex-linked disorders involve the sex chromosome. Recessive gene on X chromosome is more likely to be expressed in males. Y chromosome has no second allele that might counteract the gene on the X chromosome. Traits whose alleles are located on sex chromosomes. X-linked traits are traits whose alleles is carried on X chromosome, while Y-linked are traits whose alleles is carried by Y chromosome. Most Y-linked mutations lead to sterility and cannot be inherited. X-linked recessive disease are caused by mutation in genes on X chromosome. Males are almost always affected because of hemizygosity of the X chromosome, because they have just one X chromosome. Females are almost always carrier. The inheritance from affected male is always to daughter and never to sons. Heterozygous females are usually unaffected because of random X inactivation. A woman who is a carrier of X-linked recessive disorder has 50% chance of having sons who are affected and 50% of chances of having daughters who carry 1 copy of mutated gene and are therefore carriers. Examples include hemophilia A, G6PD deficiency, Duchenne muscular dystrophy, diabetes insipidus, and others. X-linked dominant disease. Both hemizygous males and heterozygous females are affected equally, but more severe in males. Inheritance is same as X-linked recessive. Affected male to daughters but never to sons. For example, X-linked hypophosphatemia. Biochemical mechanisms in single gene disorders. They usually cause enzyme defects, receptor defects, abnormal structural function of proteins, and unusual reaction to drugs. Multigenic disorders. Genetic disorders may also be complex, multifactorial, or polygenic. They are likely associated with effects of multiple genes in combination with lifestyles and environmental factors. They don't have clear-cut pattern of inheritance and do not follow Mendelian inheritance pattern. There is a strong environmental component to many of them. Those include asthma, autoimmune diseases, cancers, diabetes, heart diseases, and others. Thank you.